Hi everyone, thanks for checking out this video. So in the last couple of videos, we covered the format of the data set for this second project we're doing. And again, this is just a mock data set, but yeah, like I made up these numbers, but it is giving us like similar information to what a real data set for a GWAS study would tell us. So just to quickly review, we have rows for different positions in the DNA, where we have like for each row basically a chromosome number and then the actual like position in the DNA. And then at that position, there's one allele or one like single letter that a person like might have. And yeah, this one is called like the reference allele. Because it's the one that corresponds to whatever reference genome we're comparing to. But besides the reference allele, there's also an alternate allele possibility, or like another possible letter that could be found at that position besides the reference allele. And yeah, we call each one of these positions that could have the reference allele or could have the alternate allele a SNP. This is pronounced SNP, by the way. And then in this data set, we have basically samples of 200 people where 100 of them are what we're calling the normal phenotype, meaning they got bit by a zombie, but they did not turn into a zombie. They just stayed normal after being bit. And then we have 100 people who got bit by a zombie and then actually turned into zombies themselves. So we call that the zombie phenotype. And then for each of these 200 people, for each person and each SNP, we have a number here, either a 0, 1, or a 2. And these numbers tell us, like for a particular SNP, how many copies out of the 2 copies of this chromosome position they have were the alternate allele. Because remember, we have 2 copies of each chromosome. So there's 2 copies of this chromosome position in each person. So they could have 0, 1, or 2 copies of the alternate allele on their chromosomes, basically. If they have a 0 here, that means they had 0 copies of the alternate allele, meaning that both of their chromosome copies had the reference allele in this position. If they have a 1, that means they had 1 copy of the alternate allele, which means the other copy must be the reference allele. And again, simplifying assumption for this data set is that there's only 1 possible alternate allele in each SNP position. That's in real life, that's not always the case. There could be a position with like multiple different alternate alleles. But yeah, simplifying assumption for this data set is that there's only 1 possible alternate allele in each position. So if they have a 1 here, that means they have 1 copy of the alternate allele. So that means the other copy must be the reference allele. And then if they have a 2, that means they have 2 copies of the alternate allele. Both of their chromosomes have the alternate allele in this SNP position. So yeah, that's just a quick review of the previous couple of videos. This video is going to be starting to get into some of the code that we're going to use to approach this data set and like actually do this analysis. So basically our approach for this second project is going to be a bit similar to our approach for the first project. So basically the goal is going to be to go through and check each SNP one by one, like go through and like iterate through all of these rows. And then for each individual SNP, we're going to ask, okay, do we see a skew in these numbers and the labels of whether someone is normal or a zombie? And again, we call these numbers the genotype. And then we call whether someone was normal or a zombie the phenotype. And then, yeah, for each SNP, we want to ask, okay, do we see a skew between the genotypes and the phenotypes, where it looks like there might be a pattern where particular genotypes, meaning whether you have like 0, 1, or 2 copies of the alternate allele, make you like more or less likely to have a particular phenotype in terms of whether you're normal or a zombie. So yeah, we're basically like scanning through all of these SNPs and then asking this question of like, okay, does the genotype for this SNP have any impact on your phenotype in terms of whether you become a zombie or stay normal after being bit by a zombie, basically. Okay, so with that being said, I think we can start to get into some of the code now. So just to remind you guys, this code is available on my GitHub. I'll put a link to that in the description. Yeah, it's available on GitHub. I like to like teach from these Jupyter notebooks, but if you prefer just like a .py file, then that's also available. You know what? In fact, I'll show you guys quickly where it is on GitHub right now, just in case you need help finding it. Yeah, so if you go to my GitHub, github.com/Mike Saint-Antoine, just like very quickly, and I'll put the link on in the description too. It's just comp bio tutorials, intro to bioinformatics, project 2. And then this file that I'm teaching from is this one, genomics.ipynb, the Jupyter notebook. And then this one, the .py file, is the same code, but just in a regular Python file, not the notebook. And then the data, if you need to grab this data set, it's right in here, example data.csv. Yeah, you can download by clicking and then going to these 3 dots up here and then just clicking download. But yeah, so that's up on GitHub if you want to download it and follow along. So yeah, let's get into it now. First things first, I will give the disclaimer, similar disclaimer to the one I gave for the code for the first project, that the purpose of this project is to be educational. And so for this like educational purpose, I am making some simplifications. And one of the simplifications is that if you were doing a GWAS, which is the name of this type of analysis, genome-wide association study, GWAS for short. If you were doing a GWAS in real life, probably you would not want to like write all the Python code from scratch like we're going to do in this project. Probably if you were doing this for real as part of like a real life research project, you would want to use like one of the standard software tools for doing GWAS. One of the most common ones is this tool Plink. So yeah, Plink is one of the ones that yeah, it makes it easy for you to just like do GWAS very easily. It's a command line tool that you can use, and it basically just like does all this stuff for you. Basically, it makes it very easy. So yeah, this is one of the real life tools that people use in like real research projects. But even though tools like Plink are like very easy to use, and they make it super easy on you because they basically handle everything for you. They're very easy to use, but I think that actually makes them like not so great for learning these concepts. Because at least for me, when I'm like learning one of the concepts, I prefer to actually learn by doing it myself, like as much from scratch as possible. So that's why for this like educational project, I'm going to be showing you guys how to do like a simplified version of a GWAS from scratch in Python, basically, so that we can like learn how things are actually working. But yeah, if you're doing it in real life, you might want to use Plink or one of the other software tools. And yeah, so besides that, I'll also just give the like quick disclaimer that I have made some simplifications for this project to make it easy on us for educational purposes. So yeah, like I said, this is just like a mock data set that I made up, and I made it a bit easy on us. Yeah, because all of the SNPs only have one alternate allele. And yeah, just some other things too. We won't be dealing with things like covariates and additional complications. Yeah, so we're basically doing like a simplified version of this type of analysis, but I think that's okay to do for educational purposes to try to understand these concepts. And yeah, so just at the start, just import some things up here at the top. Hopefully these libraries aren't too obscure. This one's statsmodels.api. This one might be a little bit unusual, but hopefully you guys can find it and get it installed if you don't already have it. And okay, so I think like before we actually start reading in our mock data set and analyzing it, I thought I would give you guys like a brief tutorial on logistic regression. So this is a type of statistical method that we're going to be using to analyze this mock data set. And yeah, so I did put optional here. Oh, whoops. Yeah, I did put optional, and the reason I put optional is because this video is actually going to be going like a little bit in depth on how this statistical test logistic regression like method works. Basically, I'm going to be going like a little bit in depth on like what this method is and how it works. And I think it's good to know this stuff, but it's actually not really completely necessary for the purposes of this analysis to know like how it actually works. The important thing to know for the purposes of our analysis that we're going to do is that this logistic regression method will allow us to get a p-value for each SNP. And if you guys remember from the first project and from our like review of statistics, a p-value basically tells us the probability of seeing a result S extreme as the one we're seeing by coincidence alone if nothing interesting is going on, which we call the null hypothesis in statistical jargon, basically. So the null hypothesis will basically be that there's no underlying relationship between the number of copies of the alternate allele a person has for each SNP and whether they become a zombie or not upon being bit by a zombie. And basically the idea of the analysis is to do a statistical test for each SNP individually and calculate a p-value that tells us basically the probability of observing a skew between the genotypes and the phenotypes S extreme as the one we're seeing by coincidence alone if there were not a true underlying relationship between the genotype and the phenotype. And we're going to go through and use a logistic regression test on each SNP individually to calculate a p-value for each SNP that we can use to hopefully identify SNPs where there's a skew between the genotypes and the phenotypes that we would be unlikely to see due to coincidence alone, if there were not actually an underlying relationship. And so yeah, that's like the main conceptual thing to understand is just like the purpose of getting a p-value here for each SNP and like what the p-value tells us. So with that being said, like that's the important thing to understand. And if you guys don't want any additional information on how this test works, you can go ahead and skip to the next video in the series because yeah, that's like the important thing to know for our analysis. But if you want some extra information, in this video, I'm going to try to tell you guys some extra information about how logistic regression works and like basically how we get this p-value for each SNP using logistic regression. And the one thing I'll say first is that in addition to watching this video, or maybe even instead of watching it, there's a great video series by StatQuest where he really does a great job of like conceptually explaining how logistic regression works and goes even much more in depth than I'll go on this video. Yeah, like to be honest, he does a much better job of explaining it than I'm probably going to. So yeah, I would even maybe recommend watching this video series instead of my video, because I actually do think he explains it better than I'm going to explain it. Although I'm going to try. I'm going to do my best here to explain things as clearly as I can. But yeah, I do have to say this guy explains things like very, very clearly and very intuitively, and I do think he's better at explaining this stuff than I am. So I would maybe even recommend checking out his video series, like maybe even instead of this video that I'm trying to make. But with that being said, I will try to make an effort here and try to explain things to you guys. And yeah, I'm gonna try to do my best. And hopefully explain how this logistic regression test works basically. Okay, so first things first. Just as an example, we're gonna make up some test data for one SNP basically, just to work with. So this top first block of code here is just making up some test example data for one particular SNP. So it would be similar to one individual row in our mock data set. We're just going to make up an example row of data just so we have something to work with to demonstrate this logistic regression method. So yeah, don't worry too much about this stuff. This is basically just making up an example row of data to work with, where like the real thing, we'll have 200 samples, meaning 200 people. Yeah, don't worry too much about this stuff, but yeah, just some of them will be what's called cases, which for our project means the zombies, basically, people who became a zombie after being bit. And then the others will be controls, which in our case in our project means people who got bit but did not become a zombie. Yeah, but this terminology of case and control is more general terminology for how we could talk about different phenotypes in a GWAS basically. So yeah, this is just making up an example test row of data for one particular SNP, and then yeah, I like to just print things out, do some sanity checks. So yeah, we have 200 samples total, 200 people. I just printed out the first couple of the genotypes. So yeah, this is just one particular row where it's full of zeros, ones, and twos. So I print out the first couple, happen to only be zeros and ones, but we can see based on the unique values, there are also twos in this row of data basically. And then also printing out the labels where we're basically, rather than labeling them with strings that say control and case, we're just going to label the control and case phenotypes with zeros for the controls and then ones for the cases. And yeah, so in the labels array that we also have zeros and ones, but they mean different things than the genotypes. So the genotypes, the zeros, ones, and twos mean how many copies of the alternate allele the person had for this chromosome position, this SNP. But for the cases, we also have zeros and ones, but they mean something different. They mean whether the person was zero, a control, or one, a case basically, or just normal or a zombie in our mock data set. Okay, so now we can make a plot to visualize this row of data that we're working with, this row for this particular SNP. Okay, so this plot is basically showing on the horizontal axis the genotype, whether basically for this single example row representing the row for one SNP, it basically is plotting on the horizontal axis the genotype 0, 1, or 2, which again, remember, just means how many copies of the alternate allele the person had in this SNP position, basically. They could have 0 copies of the alternate allele, which means that both of their copies were the reference allele, or they could have had 1 copy of the alternate allele, which means the other copy was the reference allele, or they could have had 2 copies of the alternate allele, which means they didn't have any copies of the reference allele. So yeah, we're just plotting those. I added some what's called jitters to it, which basically just makes it easier to visualize the points. But this is not a continuous measure. Like this dot here and then this dot over here both had genotypes of 0. The jitters are just to make it easier to visualize how many points are at each position basically. But yeah, these all had a genotype of 0. These all had a genotype of 1. Same with these up here, and then these ones all had a genotype of 2. And then the vertical axis is plotting whether someone was a case or a control, which for us means like whether they were a zombie or normal. But it could be just any phenotype. Or I mean, not technically not any, because some phenotypes are continuous, but any like true false phenotype we wanted to talk about, whether something was true or false, whether they were a case or a control. And so yeah, this is a nice plot to just start like visualizing what things look like for like one particular SNP. So yeah, if we imagine again just like this SNP example being like one particular individual row, this plot lets us see like visually see all the information we're working with basically. So we can see like each of the possible genotypes, and then for each one of the genotypes, whether the people who had that genotype were a case or a control. So like a zombie or normal. So we can see like all of the possible genotypes and both of the possible phenotypes, and then the different the different combinations of these genotype and phenotypes, and then how many people fit into each one of these combinations of possible genotypes and possible phenotypes, basically. So we can like just take a look at that visually with a plot like this. So yeah, hopefully that makes sense to you guys. Just like a nice way to like visualize the information we're working with for this SNP that we have. Okay. So the goal of logistic regression is basically to fit a curve to this data. Basically to like model this data with like a mathematical curve that we could draw that would fit this data that we're seeing basically. And again, this is going to like sound a little bit complicated, I think. Even the equation looks a little bit intimidating, but just to emphasize like two things. Again, just like re-emphasize, yeah, like one, this is optional. Everything I'm about to tell you guys is optional. So if any of it is confusing or you don't understand it, it will be possible to continue this project with what we're going to do without understanding all of the underlying math, although I am going to try to make an effort to explain it. But yeah, this is optional, and if any of it is confusing, don't worry, because it will be possible to do the rest of this project even if you don't fully understand the underlying math behind this this method. And the other thing to emphasize is, again, if you do want to understand it, check out the StatQuest videos, because I do think that they probably explain this concept better than I can, although I am going to try it. I am going to do my best here. Okay, so let's try to now get into some of the math. And again, just emphasizing that this is all optional, and it's good to know, but if you want to skip it, or if you don't fully understand, it will be possible to do the rest of this project. So don't worry if any of this is too confusing. But yeah, let's try to get into this equation now. So basically, this equation tries to take as an input x, which will be the genotype of a given sample for a particular SNP. So yeah, the X here is the genotype, which could be 0, 1, or 2. And we're going to treat these 0, 1, and 2 numbers as actual numbers. So this like 0, 1, and 2 will be treated as an actual number that we can plug into this equation and get something out of it. So yeah, we're plugging in like for a particular sample, whether they have 0, 1, or 2 copies of the alternate allele, we're going to plug that into this equation and then get something out of it. So the thing we're going to get out of it is this function like p of x. This is the output of this function, basically. And this output is going to tell us the predicted probability that a given sample is a zombie or is like a case in more general terms, given what we know about its genotype X for a particular SNP. Remember, everything we're talking about here is for one individual SNP, like one particular row in our data set. All of this is talking about one particular SNP that we have like a row of data for. So yeah, basically the goal of this function is to take as an input the genotype, meaning the 0, 1, or 2 copies of the alternate allele, whether they have like 0, 1, or 2 copies of the alternate, and then plug that into this math equation, and then get as an output a prediction about the probability that that person is a zombie or is a case, given what we know about its genotype. So yeah, basically that's the goal, is to use this equation to relate the genotype to the phenotype in a way that we can get a probability estimate on whether someone is a zombie or normal, if we don't actually know their true label, but we know their genotype. So if we have like a new sample that is where we don't know if the person is a zombie or like if we know that they got bit by a zombie, but we don't know if they turned into a zombie or not, can we predict if they're a zombie or normal based on their genotype using this equation, and get some like probability on whether or not they'll turn into a zombie after being bit? Or in general terms, if we just want to know if a sample is a case or control, if we don't know the true label, can we plug in their genotype into some equation and then get a predicted probability on if they're a case or a control based on their genotype? And the way we're going to try to do this with logistic regression is by fitting a curve to the data here with this equation, where the curve will basically be plotting again the genotypes, and then plotting on the like vertical axis the probability that someone was a case or control based on their genotype. And we're going to try to get that curve by fitting these two parameters, beta 0 and beta 1. These are the two parameters that we're going to be using in this logistic regression equation, basically. It's two parameters, beta 0 and beta 1, where after we fit these to the data, then we'll have everything we need for this equation to be able to plug in a genotype X and then do this calculation with our fit parameter values, and then plug them in and then get out a predicted probability, basically. So let's try to talk a little bit about what these parameters actually mean. And again, just emphasizing again, this is possibly confusing. If there's any questions, just let me know in the comments. Possibly confusing, not totally necessary to completely understand for this project, but possibly good to know. So actually, you know what? I think a good way to try to understand like parameters in these like math models is to try to reason about it like from first principles, basically, if we can. So before I tell you guys about these parameters, I'm going to first ask you, actually, based on what I told you about like the input and output of this function, I'm going to first ask you, what is beta 0? What does beta 0 tell us? So yeah, maybe you want to like pause at this point and try to think for yourself, what is beta 0 and like what does beta 0 tell us about what we're interested in, basically. So maybe you want to like pause and try to think about it for a second before I explain. Okay, so the way to reason intuitively about the interpretation of beta 0 is, first of all, to notice that beta 0 is not multiplying X. It's not multiplying our genotype. So beta 0 basically is a parameter we have that tells us something about the probability of being a case or being a zombie, and it's independent of the genotype variable. So it's not being multiplied by the genotype variable X. So that suggests, I think, that it's telling us something about like a baseline probability that a sample is a zombie. But then there's the question, though, is like the baseline for who? The baseline for which group of people? And this is a little bit tricky. So you know what? I'll ask you guys first. Again, if you want to take a guess at this without reading ahead before I say it, maybe you want to like pause and try to reason about this. But yeah, I'll put this question to you guys. Like beta 0 tells us something about the base rate probability that a sample is a zombie, but the base rate for who? The base rate for which group of people? So maybe you want to pause again and try to reason about this and see if you can figure it out. Okay, so it is a little bit of a trick question because it is not the base rate for the entire population of people. It's not the base rate for the entire population of samples, meaning people of all genotypes. In fact, it's actually a base rate for people who have the genotype X equals 0 or 2 copies of the reference allele. And the way to see that in the equation is to see that basically if we have a genotype of 0 or X equals 0, then the beta 1 parameter will be eliminated basically, because yeah, with a genotype of 0, then we're basically multiplying beta 1 by 0, which will mean that like beta 1 will basically be eliminated from the equation for people with a genotype of 0, because it becomes just beta 1 times 0. So whatever value, like whatever fit value beta 1 had, it doesn't really matter for people with a genotype of 0, because whatever fit value is there is just multiplied by 0, and it just is eliminated from the equation for people with a genotype of X equals 0. So for people with a genotype of X equals 0, we're left with only beta 0 because beta 1 has been eliminated. So we're left with only beta 0 here in the equation. So that means that beta 0, the interpretation of this parameter is that it tells us something about like the base rate of the probability that someone is a case or not, specifically for people with the X equals 0 genotype or the 2 copies of the reference genotype. Because for that genotype, beta 1 is getting multiplied by 0 and then disappearing, and then we're left with only beta 0 to calculate the probability that a sample is a case if they have the genotype X equals 0. And then to see a specific example, if you guys want to like try it out on paper for yourself, we can see that a value of beta 0 equals 0 corresponds to a baseline probability of 0.5 that a sample with genotype X equals 0 is a zombie or is a case. So we can see that if we have someone with a genotype of X equals 0, so 2 copies of the reference, and we have a beta 0 value of 0, then we just plug in 0 for X and plug in 0 for beta 0. Then we get this whole exponent basically just being 0 plus 0 times beta 1, which is 0. No matter what the value of beta 1 is, the whole top of the exponent just becomes 0. So we get the probability equals 1 over 1 plus e to the 0 power, which remember, anything to the 0 power is just 1. So this becomes 1 over 1 plus 1, which equals 1 over 2, which equals 0.5. So basically, if you have a beta 0 fit value of 0, that means that if someone has a genotype of 0, then their probability of being a case or being a zombie is 0.5. And then a value of beta 0 other than 0 corresponds to a baseline probability of something other than 0.5 that someone with a genotype of X equals 0 is a zombie or is a case. Again, possibly confusing. Again, just don't. If this is confusing, you can either leave a comment and I'll try to explain further, or check out the StatQuest videos if you want to understand this stuff. But yeah, if it's confusing, don't worry too much, because all this stuff I'm telling you guys right now will actually not be needed to do the rest of the project. So this is just like basically extra optional information for you guys about how this test actually works and like what these parameter values actually mean. But yeah, it is optional for our purposes. Okay, and then the next parameter in this model is beta 1. And then again, I'm gonna ask you guys to like maybe pause and like think about this. Try to like reason about it yourself of like what beta 1 means and how we interpret it. Okay, so since beta 1 is the parameter that's being multiplied by our genotype variable x, the way to interpret beta 1 is basically it's a parameter that's related to how the probability that a sample is a zombie or a sample is a case, how that probability changes from the baseline, where remember the baseline is for people with genotype 0, how that probability changes from the baseline if the person has either 1 or 2 copies of the alternate allele. So basically, how does the probability that someone is a case or is a zombie change from the baseline of genotype 0 if we have a genotype of 1 or 2 copies of the alternate allele? So yeah, we can see that because beta 1 is multiplying x, which could be 0, 1, or 2. So this allows for the probability to change if we have a genotype of 1 or 2, basically. So what does it mean if we have a beta 1 value of 0? Again, I'll put that to you guys. Maybe you want to like pause and think about it. What does it mean if beta 1 is 0? And what does it mean if beta 1 is something other than 0? If you guys want to like pause and think about it. So if beta 1 is 0, if our fit value of this parameter is 0, that means that having different genotypes doesn't actually change the predicted probability. So if beta 1 is 0, that means whatever we have for x, whether it's 0, 1, or 2, it doesn't matter, because even having a 1 or a 2 will just be getting multiplied by a 0. So this whole term will just become 0. And then basically the predicted probabilities that someone is a zombie or someone is a case, if the genotype is 1 or a 2, will be the same as the baseline probability of the people with genotype 0, because the 1 or 2 for x won't actually matter because x is just disappearing, getting multiplied by beta 1, which is 0. But then if beta 1 is something other than 0, that means that if we have a genotype of 1 or 2, that means that a 1 or a 2 will change our predicted probability from the baseline. Like basically the genotype actually will matter. And by the way, in this plot, we can kind of already see that it looks like the genotypes of 1 and 2 make it like more likely to be a case, but this could be going in the opposite direction too, just so you guys know. It could be the case that for a particular SNP, having 1 or 2 copies of the alternate allele makes someone less likely to be a case. So it could be that the pattern is reversed, where maybe the genotype of 0 is more likely to be a case and the genotype of 2 is less likely to be a case. So the pattern could go in either direction, basically. Okay, so this next thing to talk about here, now that we've covered some of this like equation, is basically drawing some example curves of this logistic growth equation we're talking about. So basically, yeah, this whole block of code basically is just defining this equation as a Python function that takes in x, which is our genotype, and then beta 0 and beta 1 as the inputs, and then returns the predicted probability that a sample is a case or a control based on the genotype. Oh, also just to quickly say, and I apologize for not saying this before when we're talking about this P of X output. This P here is not the same thing as the P value of our statistical test that we're going to be eventually getting. So yeah, both this and the p-value of the statistical test are both probabilities, but they're probabilities of different things. This here is the probability that a sample from a given genotype is predicted to be a case or a zombie. And then the p-value from the statistical test is the probability of observing a result as extreme as the one that we observed by coincidence if the null hypothesis were actually true. So yeah, they're both probabilities, but yeah, this here is not the p-value of the statistical test that we'll be eventually getting. So I just want to quickly make that clear. But yeah, so this plot here just showing some example logistic growth curves. One thing to notice is that it's very similar to our previous plot of the data points that we have data for. So the horizontal axis is actually the same, just plotting the genotypes 0, 1, and 2. And for our plots of our curves, we are plotting them in this continuous way where we can see that there is stuff being plotted in the middle in between 0 and 1 and then 1 and 2. But in terms of biological interpretation, even though the curves are continuous, in terms of interpretation, the only input values of X that we're really interested in are the 0, 1, and 2 values for our 0, 1, and 2 genotypes. So even though we're plotting them to be continuous, in that there is space between 0 and 1 and 1 and 2 that's being plotted in the curve, the interpretation of them is still discrete in the sense that the only things that it would make sense to plug into them to get a value out would be 0, 1, or 2. And then the vertical axis is very similar to what we were talking about before with this plot, except in the plot before, the vertical axis was plotting just 0 or 1, whether someone was a control or a case, and then plotting actual data points where we know if the person was a case or control, and then plotting them all at 1 or 0. Where these are actual data points where we know if they were a 1 or a 0, and we're plotting them accordingly, basically. On the other hand, for these logistic curve plots, 1 and 0 still mean the same thing, where 1 means case and 0 means control, but we are interested in this intermediate continuous space of probabilities between 0 and 1. So the interpretation is very similar to the interpretation of the previous plot, except instead of plotting whether someone is actually a case or a control, we're plotting the predicted probability that a sample with a particular genotype is predicted to be a case or control. So now the vertical axis is actually plotting something continuous. It's plotting the probability that a sample is a case or control, and how that relates to the genotype of the sample. So these are probabilities here, where maybe for a sample with a genotype of 0, based on whatever model we have, whatever fit parameters we have, it could be predicted that the sample with a genotype of 0 is maybe 0.9 up here maybe, or 0.5 or 0.6 or 0.2. So this now is a continuous probability being plotted on the vertical axis here. And again, the goal of everything we're going to try to do with the logistical regression method, basically find the particular values of beta 0 and beta 1 that result in a curve that most closely matches the data that we have. So we want to find the values of these beta 0 and beta 1 parameters that give us a curve that most closely matches the data that we're fitting to, basically. And the way this is done behind the scenes is with something called a maximum likelihood method, basically. And if you guys want to learn more about how that works, There's again like a very good there's a very good like StatQuest video on it. But for now, we don't have to worry too much about how the fitting is actually done, because the stats models package in Python will basically take care of that for us. So this next block here is how we actually do the fitting of the logistic curve to the data, where again we have our we have our like SNP data that we made up for an example, and then we can go ahead and fit that model to the data, where again we have our SNP and our labels from before, and fit it to the data, and basically get this whole thing is like a whole big result of this logistic regression model basically. So yeah, this block of code here is just fitting the model to the data using the Python stats models package. So yeah, a lot of extra information here, a lot of stuff we don't actually need for our purposes, but the important things are that after we do this fitting, we can go ahead and like pull out the fit values of beta 0 and beta 1 from our results basically. So basically for our example SNP data, after we do the fitting, we can pull out the values of beta 0 and beta 1 that cause the model to most closely fit the data, basically. So we can pull those out and then print them. Yeah, these are our fit values of beta 0 and beta 1. As a quick side note, just a little note on terminology. We've been talking about these parameters of beta 0 and beta 1, and as a general convention, when we're talking about actual fit values of beta 0 and beta 1 that we get by fitting a model to data, we'll then talk about those fit values with the notation beta 0 hat and beta 1 hat, where the notation looks like this, and they're pronounced like this thing at the top is pronounced like hat, basically, because yeah, it looks like it's like wearing a little hat. Yeah. So yeah, we'll call these like beta 0 hat and beta 1 hat, or even more simply just like fit beta 0 and fit beta 1. But we will distinguish between those fit values of beta 0 and beta 1 that we get by fitting the parameters and like fitting the model to the data. We'll distinguish between those like fit values versus the actual parameters beta 0 and beta 1 that we can think of as being like true underlying values of these parameters, like in the universe, that we can try to estimate with this fitting. So we can like try to estimate them with beta 0 hat and beta 1 hat that we get after the fitting, but we'll still think of like beta 0 and beta 1 as just being these like true underlying parameters that we can estimate with these like fit values of the parameters, but we can never really be entirely sure about. So yeah, it's like we have the fit values of these parameters, and then we have the true underlying values of these parameters that we're trying to estimate, but we can never be truly sure about. So yeah, there's like a little conceptual distinction there. But yeah, once we have our fit values of these parameters, we can then go ahead and basically like make again this plot of our data points, but now actually plot the logistic growth curve that we get using these fit parameter values. So yeah, basically we now have a fit curve that tells us like, okay, we fit this curve to our data. So now this curve tells us like, okay, if we get a new sample and like we know that someone has been bit by a zombie, but we don't know if they're going to turn into a zombie or not, or if we just have a new sample where we know the person was bit by a zombie, but all we have is their genotype, we don't, and really their genotype for only this SNP, we don't know if they were if they actually turned into a zombie after being bit or not. If we don't know the true label, we can say, okay, we have this logistic growth curve that we fit to the data, and now we can say, okay, we can try to estimate the probability that this new sample is a zombie or normal based on their genotype for this SNP. using this model that we fit to the data where we knew the genotypes and labels for a bunch of people. So we can then try to like plug in whatever genotype this new sample has. Maybe it's a 0, and we plug it in and we say, okay, we can predict like a pretty low probability that this person will turn into a zombie after being bit. Or if they have a genotype of 2 for this SNP, maybe we predict a pretty high probability that they'll turn into a zombie after being bit. Or if they have a genotype of 1, maybe like an intermediate probability. And then, yeah, this next block of code is just going ahead and just actually calculating the probabilities by plugging these like genotypes of 0, 1, and 2, as well as the fit model parameters of beta 0 and beta 1 into our actual Python function of the logistic growth equation. And just seeing like what the predicted probability would be using these fit parameter values for each of the possible genotypes that someone with that genotype for the SNP would have the zombie phenotype after being bit by a zombie. Okay, so last thing to get out of these like model fitting results for this SNP. And last thing, and really most important thing for the purposes of this project. In fact, the only important thing that we really need to actually need to know is the p-value. The p-value for like the relationship between the genotype and the phenotype for this particular SNP. And we can go ahead and grab that with this line of code here. So this is giving us the p-value for the relationship between the genotype and the phenotype for this particular SNP. So the technically correct definition for what this p-value means, like for the logistic growth model, is that it's basically the probability of observing a fitted value of beta 1, which again is the parameter that multiplies the effect of the genotype. Basically, the probability of observing a fitted value of beta 1 at least as extreme, meaning at least as far away from 0 as the one we obtained from the model fitting, measured in standard error units from 0, due to like randomness, due to random chance of the random sampling, if the true underlying parameter of beta 1 were 0. So basically that's the technical definition of the p-value for the beta 1 parameter in the logistic regression model. So like again, if the true underlying value of beta 1 were 0, that would mean there's no underlying relationship between the SNP genotype and the probability that someone with the SNP genotype is a zombie. So, but again, like the true underlying value of beta 1 is conceptually different from our fit value of beta 1 that we get from fitting the model to a sample of people with like the labels and the genotypes. So the technical definition of a p-value for this model is basically asking like, okay, we have our fitted model of this parameter, and we want to say, okay, if the true underlying value of this parameter were 0, what would be the probability of getting a fit value as far away from 0 as the one we got by coincidence if the true underlying value were 0. That's like the technical definition for the logistic growth model. But for our purposes, I think the easier to understand, like sloppy, like less technically correct, but easier to understand and intuitively definition of this p-value is it's the probability of seeing a skew in the data between the SNP genotype and the zombie phenotype, or just the case phenotype, by coincidence if there were no true underlying relationship. That's the important part for our project here, is the p-value quantifies the probability of seeing this much of a skew. Like we can literally see the skew in the data points here between the genotypes and the like the case status seeing this much of a skew between the phenotype and the genotype, by coincidence, if there were not actually an underlying relationship between the genotype and the phenotype. Probability of seeing that by coincidence, where again, a lower number means less likely to see by coincidence, which means possibly more likely that there is actually like a relationship between the genotype and phenotype. Okay, very last thing, at least for this video. And I'm going to try to make this fast because I've realized this has been a long video, and if you guys have stuck through it all this way, even on this optional video, then I'm already proud of you guys for sticking with this. So yeah, I'm going to try to make this like last part like extra quick, just like one final last thing before we close out this this video. But yeah, one final last thing to note is that for this project, we're basically doing like a simplified analysis where for each sample, we only know the label of if they were a zombie or not, and the genotype for each SNP. We only know the genotype and the zombie status or the phenotype. And so yeah, I purposely made this like a simplified data set to make it easy on us. But if this was a real life GWAS analysis, we would probably have more information. Like we would probably have like the person's like maybe age, sex, ethnicity, health background, etc. More stuff like that about each sample. So we might try to model then the probability that someone was a zombie or not based on more information than just the genotype for each particular SNP. We might try to incorporate this other information about this other what's called like covariates that we have. They're like demographic information, their age, sex, ethnicity, their like health conditions, stuff like that. We might try to also incorporate that information. In fact, that is in real life, that's like an important part of the GWAS. And another nice thing about these like real software packages like Plink is that they make it like easy to do that actually. So yeah, we're like, this project is going to be a simplified project where we won't worry about this stuff. But if this was a real thing, like a real analysis, a cool thing about the logistic regression model is that it lets us easily add in more variables to try to predict the phenotype besides just the genotype. So we could easily define more variables and just put them up here in this exponent that we're exponentiating E with. We can add more parameters here that are like multiplying more variables. So maybe, for example, yeah, like maybe we want to add in two more variables, y and z, where maybe y is a 0 or 1 and tells us whether the person was a smoker or not, where like 1 means that they were a smoker and 0 means that they were not a smoker. So maybe we add in y, and then maybe we add in another parameter, beta 2, which multiplies y and then tells us the effect of the smoker variable on the probability that they were a zombie or not. And then maybe Z is the person's age in years, and maybe Z just yeah, we add in Z to the model where Z tells us the person's age. And then maybe beta 3 is a new parameter that multiplies Z and tells us yeah, like whether the person's age had an impact on whether they became a zombie or not after being bit. And then yeah, so I just put another block of code, just again like fitting the model with these extra parameters. And then we could also like estimate the effects of these extra parameters too. And just one final thing to close out on, another question for you guys. Like I've been telling you guys like how to reason about this equation like intuitively. So another question for you guys before we close out, and just something to pause and think about. Something to think about is like how do these extra parameters change the interpretation of beta 0? Do they change the interpretation at all about what beta 0 tells us? So maybe you guys can like pause and think before I tell you the answer. And the answer is yes. Again, this is kind of a trick question because yeah, like adding these new parameters does actually change the interpretation of beta 0. Because if you guys remember from before, beta 0 was telling us about the baseline rate for someone with a genotype of 0, which would cause beta 1 to be canceled out. So now again, now beta 0 would tell us about the like baseline rate of the like baseline predicted probability that someone was a zombie if all of these other variables were also canceled out by equaling 0. So now beta 0 would tell us something about the baseline rate that someone was a zombie if they had a genotype of X equals 0, a smoker status of Y equals 0, meaning they were not a smoker, canceling out any effect from beta 2, and then an age Z of 0, canceling out beta 3. So in this case, in this example, like the age of 0 doesn't really make sense, but we could possibly, if we wanted to, define age as being like normalized around some like mean age or something, so that Z equals 0 would maybe mean they just have the mean age. But yeah, so just to emphasize, like beta 0 tells us something about the baseline rate of the predicted probability that someone is a zombie or is a case if all of these other parameters were 0 basically. So that they cancel out the effects of these other parameter values. Okay, so that's everything I have for you guys for this video. Again, if you made it this far, I'm proud of you. I realize this was a long video, getting into the weeds of some of this math. Yeah, proud of you guys for sticking with it to the end. Again, all of this was optional, so if anything was confusing, you can leave a comment and I can try to explain further. Or yeah, like this is all optional. If you don't understand all of the math, the important thing to understand is really just the interpretation of the p-value. That's the only thing we're going to really need to be able to apply this statistical test to each one of our SNPs in the data, and then get a p-value for each one to look for the SNPs that might have a relationship between the genotype and the phenotype. And this math stuff was just like kind of extra stuff, basically. But yeah, thanks for watching, and I'll see you guys next time.